The overall goals of this analysis are to determine the cellular basis of maize leaf growth and to use these data to investigate molecular growth regulatory mechanisms by performing developmental stage-specific sampling strategies.
This method can help to address a key question in plant developmental biology. How do cell division and cell expansion contribute to differences in whole organ growth?
This approach allows the investigation and spatial mapping of biochemical, molecular and physiological processes underlying the growth of the whole plant organ.
Though this system can provide insight into maize leaf growth, the technique can be used for other systems, including other grass species and organs.
Visual demonstration of these methods is critical, as the harvesting, sample preparation and measurement steps are difficult to perform without a demonstration.
Demonstrating the procedure will be Katrien Sprangers and Viktoriya Avramova, two PhD students from my laboratory.
To perform a full kinematic analysis of leaf growth in monocots, grow at least 15 plants for each treatment and genotype under controlled conditions in a growth room. When a leaf of interest appears, use a ruler to measure the length of the leaf, that is, the length from the soil to the tip of the leaf, daily until the leaf is fully expanded. At the developmental stage of interest, cut the above-ground part of the plant from at least 5 representative plants as close to the roots as possible to keep the meristematic region intact. Then, starting from the outer leaves, gently unroll all of the leaves up to the leaf of interest one by one, removing a few extra millimeters from the base.